Good morning, good afternoon, and good evening, depending on where you are in the world. I'm delighted to welcome you to the latest BMKGENE webinar, How to Start Your First Spatial Transcriptome Study. I'd like to thank you all for taking time to attend today's webinar. And today's webinar is released for around 40 minutes, with 5 minutes for the question and answers at the end. And you could submit your question via the Q&A button at the bottom of your screen. And we are looking forward to receiving your questions. And we will share the slides in a recording of webinar via email. Now, I'd like to introduce the speaker of today's webinar, Lin Li, who's our senior product specialist of the Global Service Department. Lin owns years of experience in high-throughput sequencing area, and she got her doctoral degree in University College Dublin, Ireland, during which is her main research interest was on bio-nano-immunology interaction in terms of gene expression and genome-wide histone modification profiling. After that, she joined Biomarker Technology as our senior product specialist of Global Service Department. In the meantime, she is also involved in our in-house research projects, such as development of long-read sequencing-based genome component predication tools as a postdoctor in BMK Research and Department. In this webinar, she will share the basics and principles of spatial transcriptome sequencing technology, like how to start a spatial transcriptome study and what you could expect from your spatial transcriptome data. Hi, Lin, it's over to you.
Okay, thank you, Emma, for the introduction, and welcome everyone for joining our webinar today. And I'm really happy to be here to share my experience in giving a spatial transcriptome project or study. So in today's webinar, I will mainly give a general introduction on spatial transcriptomic technologies and also how the studies looks like. And also I would like to take our service workflow as an example to show you how we run a spatial transcriptome study. And I will also couple with a case study to make a better understanding of what we are working on. So before we go into the details of spatial transcriptomics, I would like to have a very brief recall on the typical mRNA study technology, which is NGS-based mRNA sequencing. It has been widely applied in most of the biomolecular level study. And in this most typical technology that we used, if we're thinking of the way that we prepare the samples, the thing we do is that we have one sample in the tube, and whatever in this tube will be lysed in the lysis buffer, and we take the total RNA from there. And from that material, what we can get is an average gene expression value for each single gene and in each single sample. So in this case, for this type of technology, we are really taking average of a big population of cells or whatever. And we all know that in biology studies, taking average is a very, very risky thing to do, because some of the samples that we are having is kind of heterogeneous, or like one number is not good for representing the total biological status. So there is another technology coming up, which is single cell mRNA sequencing, and enables us to have a gene expression profile for every single cell. So this can help us answer more biological questions, especially on the composition of a very complex tissue. Still, like when we do the single cell mRNA sequencing, the first step that we are doing is to dissociate the tissue sample into single cell suspension. So there is information missing here in this step, which is the spatial content. And the spatial content actually is very, very important in many of the biological arenas. So the spatial transcriptomic studies help us to give this gene expression profile, and at the same time, it saves the spatial content for us. So I also give an example of different how spatial organization can be very important in most of the biological areas. For example, in this oncology studies, especially for this tumor microenvironment studies, there is a calling for knowledge of the original environment of cells. Like the example that I showed here is looking at the tumors and the interface of the tumor with other tissues. And also it's like in oncology, the spatial organization is very important for looking at different disease progress, like cancer pharmacology and immune infiltration as well. And in neuroscience, we know that, for example, brain is a highly heterogeneous and complex structure, and the spatial organization of brain can give us more information in looking at the organization of the neural system, and also in some studies when we want to decouple the normal and damaged part of the tissue in a disease, this is also very important information that we want to keep. And another example here is the developmental biology, where we need to tracing the development and differentiation of the cells in context of the tissue structure. And here I put an example on the development of heart tissue here. And they have this profiling on the cellular differentiation and the morphogenesis and etc. And in the immunology part, I also give an example here, which is looking at the immune landscape in the inflammatory tissue, where it's very important for us to track this activity of immune cell activation and also in context of spatial information. And the last example I gave here is because not only for like human or other remodeling studies, in plant studies, it's also very important when we are looking at different angle of this area. For example, this is a study that BMKGENE also contributed in. It just published, I think it's in September this year, and it's working. This is the first study on looking at the flowering, the flower development of a plant. And the spatial transcriptome sequencing really helps us to have more information in this type of studies. So this is how we would like to move on to the spatial transcriptome sequencing. And here, if we look back into the history, the foundation of current era spatial transcriptome technologies were built since like 1970s, it's long ago. And like over the years, they're kind of getting upgraded a lot for different versions, and there is explosive growth, especially in the last 10 years or so, just taking advantages of the rapid development of the sequencing technology and also the imaging technology. And most of the spatial transcriptome technology that we are having, or the commercially available platforms, they can fall into mainly 4 different categories based on the principles they're using. For example, the region of interest, where they use this microsectioning based methods to take the profile of the specific region that they're interested in. And also the fluorescent in situ hybridization, they link coupled with this fluorescent signal on the target probes. And in situ sequencing, although it's also called sequencing, but it's actually more like a microscope-based technology where they make use of this rolling cycle amplification technology to give us the similar information of what has been expressed in which part of the tissue. And the last one, spatial barcoding, is a bit different from the previous 3 that I introduced. This spatial barcoding is mainly based on the sequencing technology itself by giving barcodes on different spots of the capturing area, which I will also introduce later. So today, mainly, I will give taking spatial barcoding technology as an example to show you how we do the spatial transcriptome study. And So there are two main platforms that we're using in our company to do this spatial transcriptome studies. And the main one is, or the most widely applied one is the 10x Visium introduced by 10x Genomics. And here it is a figure showing us how 10x Visium helps us to save the spatial information. So this is the slide of 10x Visium where they have 4 capture areas, and each area it has a size of 6.5 millimeter by 6.5, where they have many probes on top of the capture area. And for each spot, the size of the diameter size of each spot is around 55 microliter, and the distance between center to center is around 100 micrometer. This is what we call the resolution of a spatial transcriptome study. And on each single probe, there is a series of sequences that may help us keep the spatial information as well as capturing the mRNA molecules. So the first part is the black one, the partial read. The black part is mainly to align, fit into the sequencing sequencers, for example, Illumina, and they have this common adapters to align with the flow cell. And the most important part here is the spatial barcode, where for each single spot, they have a unique barcode, where this part will be amplified with the mRNA molecules that captured by this poly-D tail. And then in the final data, we can make use of data to align back to every single spot that the barcode tells us. And there is another barcode here, it is the UMI, which is the barcode that we use to mark every single molecule. And the reason that we need to introduce UMI here is that if you imagine the size of this spot, there will be only very limited amount of mRNA released onto each single spot. So there will be many, many rounds of PCR reactions. And this UMI just helps us to have a much, much more accurate quantification of this mRNA data, expression data. So once we can release the mRNA molecules from a tissue onto this capturing area and then labeled by spatial barcode and UMI, finally what we can get is like the figure that I shown below here. Like from an HE stained tissue, we can have spots with different expression profiles, and these profiles can be used to clustered into different cell clusters or spot clusters to tell us which part is more similar to each other. So this is the basic principle of how 10x Visium runs. And here, taking 10x Visium-based service as an example. So this is the summarized workflow of this whole study. So the green block that I put here is the ones that in our service, what we need from our customer, for example, if it is OCT embedded block or FFPE block. And we will perform all the steps that I showed here. I will give more details in the following slides. And then by the end, what we will provide is an integrated data analysis report with most of the analysis data, no matter it's QC or more in-depth data mining. So 10x Visium or any kind of spatial barcoding related technology is still rather new. It's rather new technology for us. So there are still many challenging tissue types that we are still trying to overcome, or it's still very challenging that most of them they are failed. So before we really start a new experiment or new study on spatial transcriptome study, there are mainly 3 aspects that we can briefly look into before we start. And for example, the first one is the tissue type or the sample type. It is preferable for cross-sectioning. And in this part, we are mainly looking at the hardness, like the texture of the tissue, the thickness of the tissue. And for example, for hard bones, now it's still very, very challenging to work with. And if the tissue has been performed for cross-sectioning for years, or it has been done successfully on other HE staining related technologies, it's a very good start of this spatial transcriptome study. And the second aspect here that I put is the preferable for mRNA capturing. And some of the consideration here is that if this tissue type is rich in RNA And if it is rich in different enzymes, for example, the one that I listed here, the pancreas, this is rich in many different enzymes, and most of the mRNA molecules that we harvest from these type of samples, they may heavily degraded, and with a heavily degraded mRNA molecule, it's really difficult for us to capture in the downstream and do a better, have a better data in the following steps. And another consideration here is the permeabilization, and this is mainly for example, for some plant tissues, if they have this wall, it's really difficult to permeabilize, and if it is very old tissue, it's also very difficult to get the mRNA release from the cells. And the third aspect that we are considering is if it is preferable for analysis, and the main thing here is that if the species have a very high level reference genome, like chromosome level genome. So if it is a species, like some rare species, they don't have a very good reference genome, then thinking of we are only capturing a very end of the transcript, it will be very difficult to identify enough genes for every single spot. So these are the considerations. Once we find that our sample is good, like very perfect for this experiment, then the next thing is that how we prepare these samples. And the most commonly used technology or the method here is to do an OCT embedding, and it's very common in most of these tissue-related study labs. And here I just showed some images of how we do this OCT embedding, and we found that this small cassette was 7 by 7 in size. It's perfect for fitting into this 10x Visium slides, because the capturing area that I just introduced is around 6.5 by 6.5. So we need to make sure that our tissue fit in this size and it's not too small that waste most of the capturing spot on the capturing region. And yeah, that goes into the size of the samples that we are looking for. The optimal size is around 6.5 by 6.5 by 5. This is a very perfect size. And alternatively, it can be a little bit small, or if it is really a tiny part of the tissue, what we can also do is to combine two different tissues in one OCT embedded block. So that means on every single capturing slide, we have two samples fit in one. And in terms of the sample preparation, we always recommend to have backups and to have more biological backups that we can perform several rounds. And especially in our process, we need one block to do a bulk mRNA extraction and another block to do the real sDRNA experiment. And we always want a backup sample. So once the samples are ready, the first thing we need to do is to do the Spark mRNA quality control, which means we will extract total RNA from the tissue and then just send it for checking the concentration of the mRNA, the integrity of the mRNA, just make sure that we don't waste the rest of the kit or the rest of the chip. And if the mRNA quality is good enough, we can perform this tissue sectioning on our block. And the tissue sectioning part is actually we can perform it live with our customer, like either it's an online meeting or it's an audio meeting. So just to make sure that we are cutting in the right direction that our customer wants, and also we are cutting onto the right part that best represents the sample, or it can like by eye they know that they have all the tissue types that they want on this single section. So once we confirm that this tissue section is the perfect one that our customer expected, the next step is to do a permeabilization optimization step on this tissue sample. So different tissue types, they may have very different permeabilization time, which means that in this step, we will need to make sure that most of the mRNA released from our samples, released from the cells, and been captured by the probes on the optimization chip. And at the same time, we don't want to overflow the mRNA, because you see, as you can see, the distance of the spot can be very close to each other. And if we perform the permeabilization too long, then the mRNA may flow into the spots that's close to it. So this will mess up the data as well. And as you can see the example here, we want a clear edge of the fluorescent signal, and at the same time, it has a relatively good intensity of the signal. So this step is all for us to know the best permeabilization time that we can use to the real gene expression experiment. So in this experiment, once we know the time, we will do the real sectioning and have the section onto the gene expression chip. So the first thing is that we will do a HE staining and imaging, which will, in the downstream analysis, can be coupled with the gene expression data. And the permeabilization will be done according to what we have got, the information we got from the last step. And then the RNAs will be released onto the capturing area and being barcoded. And the barcoded cDNAs will be harvested and amplified and do the normal library preparation step to make a structured library where we have these adapters and a spatial barcode UMI and poly-GT part, and the real insert in the that representing our mRNA molecules. So once we have the libraries ready, normally we put it onto Illumina NovaSeq sequencer and to perform this P150 sequencing. And the data recommendation here is that we need to have at least 50,000 P reads per spot to make sure that we recover most of the information. And thinking of the coverage of the tissue on the chip can be a little bit different. So the final data can be around 30 to 60 gigabase clean data for each single capture area. And in our service workflow, normally we recommend to have a 60 gigabase data package per sample. So once we have this sequencing data, there is a standard data analysis pipeline that built in our company where it consists the raw data quality control, the basic analysis to give us the cell expression metrics, and then the downstream ones that do the data mining. And I will give you more details here. So, for the typical RNA sequencing or other sequencing results, the first thing we need to do is to make sure that the data is good enough. So there is always a data quality control part. And in spatial transcriptome sequencing, the QC has a bit more numbers that we need to look into. The first thing is that the tissue coverage, which means the number of spots that has been found under the tissue here. And another one is the data output, and not only the entire data like 60 gigabase, but also the main reads per spot. And as I mentioned, the optimal part here is that we have at least 50,000 reads per spot. And the most important thing, the most important indicator here is the gene detection per spot. So normally the number of gene detection per spot is around, say, 1,000 to 5,000, depending on different tissue types and also the performance of the experiment. And if it is within that range or a little bit larger than that range, that means it's a very good data set that can be used for the downstream analysis. And similar to the other sequencing technologies, we are also looking at the accuracy of the sequences, for example, Q30 bases in different part of the reads in barcodes, which makes sure that we have accurate location. And you can see on each edge of this square, there are some different markers here that tells the software the real localization of this chip and the tissue. And the Q30 bases in RNA reach, which means we need to have accurate readout of the RNA molecule, and also the Q30 bases in UMI gives us the confidence saying, okay, the expression is accurate. So this is the QC part of the spatial transcriptome data. And the main analysis here is that what we can get from the data from a single sample, like inner sample analysis. The first one, as I mentioned, we need to detect genes for every single spot and get the gene expression profile for every single spot here. So here I give some demo results in our analysis pipeline, where you can see the gene richness for different samples and the distribution, and also this can be marked back onto the HD staining images and tells you where are kind of more active in terms of the RNA content and where are a bit colder. And another very important thing here is that since we have this gene expression matrix for every single spot, we can do analysis like PCA analysis or other reduced dimensional analysis to make it into different clusters according to the similarity of different spots. And these different clusters can also map back onto the tissue sample to tell you, okay, this cluster is here and there is another cluster on the edge of this tissue, and they can be sectioned for many different types of cells or spots here. And the next step, once we have the spot clustered into many different clusters, we can perform this differential expression analysis to different clusters. And the way we do it is to compare the gene expression of cluster, say cluster 1 versus all the rest clusters, and then identify the marker genes for every single cluster. And this is a very typical result of this analysis, where it gives a heat map on the gene expression profile for different clusters. And you can see the red part is the highly expressed ones in a specific cluster. And these are the genes that we may be interested in if we're looking at specific region of this tissue type. And besides that, we can also have this hyperviable gene identification, where it tells you which genes are kind of jumping across different clusters or different part of the tissues. And this can be very important biomarker as well. And besides that, what we can do, so we can always play with the numbers that we get from this spot clustering or gene expression metrics. For example, here, once we have the marker gene identified, we can also plot the heat map back onto the tissue sample to see, okay, this gene has a little bit higher expression here. It also expressed in other parts, but even though they are marker genes of one cluster, but we can still find some expression in the other part. And this can be done for many different genes. So, yeah. And once we identify the genes, for sure, the next step or the next thing we want to do is to know the biological meaning of these different genes. And these are the very typical studies that we would do, like geo enrichment, like protein-protein interaction network, and KG pathways analysis, and etc. So these are just helping us to give a biological meaning of what we have identified from the comparison. And besides this inner sample analysis, if we have two different samples in one study, for example, we are looking at both healthy tissue and also the damaged tissues. In this type of study, what we can do is that after inner sample analysis, we can combine all the spots of the both samples, or if there are more, we can combine all the spots together and do a reclustering of the spots. So in this way, we can see that there are some unique clusters appeared in one or the other samples, and there are some common clusters that exist in the different part of the two samples. So this can be very important or very useful in our study on comparing different conditions or comparing different treatment or different disease stages and etc. And there are also very similar analysis. Like the inner sample one, we can identify the marker genes of different new clusters that we got from the combined spots. And these ones can be also mapped back onto different tissues to give us a better idea of where they has been highly expressed or where it has been more cold. And the differential expression analysis between samples can be done also, if there are common clusters existing in both groups or both samples, we can compare the differential expression of this common cluster, and then still you can find some of the genes that are still a little bit different, but overall they are more similar. And these genes can also be sent for this function analysis, for example, the GO or KGG enrichment. And besides all the ones that I mentioned, they are more like a pipeline analysis or standard analysis, most of the people will look into like at the very beginning of the analysis. And to get more information or to do a more in-depth data mining, there are more customized data analysis that we need to do. For example, if by this HE image, we know that there are some like specifically interested part that I want to compare between each other. We can always, for example, draw a circle here and extract the expression information here and compare with the one that we want to compare. So this can all be done very flexibly, and like it's more depending on the real research questions that you may have in the future. So another thing that I would like to emphasize is that we are talking about spatial transcriptome sequencing, and it always happens that people confused with single spot with single cells. So if you're thinking of the size that we mentioned in the previous slides, the diameter of the spot is around 50 micrometer, which means that it can contain like tens of cells in each spot, or like maybe a little bit less. So that is not exactly a single cell level study. And this can give us enough information of what has been expressed in each part of the tissue, but it still can't give us very accurate information on what exactly the cell type is on that part. So there are many studies coming up today to combine the single cell mRNA sequencing with spatial transcriptome sequencing together to like first have this single cell resolution information in terms of cell type annotation, and at the same time we still want to keep the spatial content. And the most common way to do it is just to make a correlation analysis between this cells information and spot information to give a better annotation of, okay, these spots are more likely to be one of the cell types. And this is a very simple example here. And I would like to show you a case study on here. So this is a very integrated study that they try to get this spatial temporal cell atlas of this developing human heart, and they use many different technologies. So in this study, they take the three developmental stages of this human cardiac tissue, and they have many different, for example, for this 1.5 weeks tissues, they have 4 sections. They have more sections on the 6.5 weeks, and they have some sections for 9 weeks, and they all do the spatial transcriptome sequencing on these tissue types, and then combine all the spots together to make this clustering figure. And in this cluster, they identified 10 different clusters, and once they align these clusters back onto every single tissue or every single HE staining images, they found that the clusters nicely corresponded to defined anatomical regions in all 3 parts. So for example, if you can see here, the microcardium spot, like the blue ones and the light blue ones, they are kind of shared by all stages from 4.5 weeks all the way down to the 9 weeks. And you can see a growing area along the time. And also, like if we look at the outflow tracks, you can see a more condensed signal like identified in this 9 weeks one. So if you're interested in this paper, I also provided the DOI here, where you can see there are more data or more results interpretation that they mentioned in this paper. And also what they did on the spatial transcriptome sequencing data is that they try to get the differentially expressed genes in every single cluster from cluster 0 to cluster 9, and then they put these DEGs into this geo enrichment analysis. And as you can see from cluster 0 to cluster 4, those microcardium related tissues or the spots, you can see they shared this process of the cardiac muscle growth or cell differentiation. And you can see for this compact ventricular myocardium, there is a strong enrichment in this the basic metabolism pathways. And you can see there is a new or a different cluster. This cluster 3 is also ventricular myocardium. they can be distinguished by another two pathways, which is mainly the oxidative metabolism and etc. Like they have many informations here. So this is how we make use of this spatial transcript data into a real developmental biology study. And in this study, as I mentioned, this is not a very like strict single cell level. So what they add on is to use single cell mRNA sequencing as well as in situ sequencing to give a cell type mapping of this human developing hearts. For example, in this single cell mRNA sequencing, they use this 6.5 weeks tissue as an example and cut it into 2 sessions to perform this single cell mRNA sequencing. And they combine or they correspond the they correlate the data of this single cell analysis with spatial transcriptome analysis result, and they have a very good correlation between each other. And with this single cell information, they are able to name or to annotate more accurate cell types in the clusters that they harvested. So in the single cell mRNA sequencing, they managed to get 14 different cell types in this tissue type. And to get a more accurate mapping back of this cell type onto the spatial transcriptome data, what they do is they use another technology that I mentioned in the first or the second slides, is the in situ sequencing, where they need to design several probes to validate if the spot is the right cell types that they are thinking or they're expecting. So in this study, they use 96 targeted probes, like targeting to 96 marker genes that they identified in this previous experiment. And by the end, by this, the combination of these 96 targets, and they are able to name more accurately where each cell type located in this tissue. And they have done many different sections in 3D structures. So by the end of this paper, they are introducing this 3D model of this heart developing. So this is a very good study on how to use spatial transcriptome sequencing in developmental biology. And I would like to take some time to introduce a bit on the BMKGENE spatial transcriptome sequencing services. So in our company, we have performed over 50 different tissue types, and we have processed over 1,000 samples for the spatial transcriptome sequencing studies. And I have listed most of the some of the examples here. For example, most of them, they're still on human, and there are some for mouse and rat, and some like model species like zebrafish and etc. And besides the 10x Visium-based spatial transcriptome technology, biomarker technology BeiGene has just launched our own spatial transcriptome platform or chip named S1000, and this is a subcellular-level technology where it gives a much higher resolution in terms of telling like the for every single spot. So this technology is also depending on the spatial barcoding principle, and as you can see, like we managed to enhance the resolution from 100 micrometer into 5 micrometers. So for every single spot, the diameter of the spot is 2.5 micrometer and the distance between is 5 micrometers. So this technology really helps us to have a more in-depth, more detailed information in the tissue sample, especially when we are looking at a very complex or very tiny structure alteration and etc. So so for our own spatial transcriptome chip, we also designed our own data analyzing toolkits that fits the data that we harvest from our chip, where it includes tools that can do from both from all the analysis that from sequencing data down the way to the gene expression matrix, and from here all the downstream analysis will be pretty much similar to every spatial technologies. And one of the beauty here is that although we managed to increase the resolution to 5 micrometer, and still for some of the tissue types, 5 micrometer may be too much, or it may have many noise as well if it is if it is not very rich in RNA content. So in the analysis, we are able to analyze data at different subspot levels. For example, if I know that for my cell types, for my tissue types, the perfect cell size can be around 20 micrometer, I can use the analysis at subspot level 3, or I can make it more larger. So here I would like to share another example here to show you how different resolution can make a difference in the final align or final cluster of the spots. So this is the HE staining image of this tissue, and we can see a clear edge here. And if we enlarge this part, we can see there is kind of a break in the middle. And if we like move to the 100 micrometer one, we kind of have a rough line here, but we lose some of the layers that we can still see by eye. And if we increase it to 50 micrometers, we can kind of see more layers and more clear structure. And from 35 micrometer one, we start to see a little bit break as we saw here. And but at the same time, it starts to have some noise. So it's always a balance to see if we can have a better clustering, and at the same time, we can minimize the noise. So this is the subcellular resolutions, spatial transcriptome studies that we can do at the moment. So this is just launched in Beijing, and it will be, I think it will be commercially available very soon. And for the last few minutes, I would like to give a very brief introduction on BMKGENE. Although we have provided sequencing service in China for over 13 years, but we are rather new to our European customers and also Asian customers. And basically, BMKGENE, what we are trying to do is to make the best use of the current sequencing technologies and also other platforms to answer different biological questions like genomics, transcriptomics, and single cell ones, the spatial ones that I mentioned, and metagenomics, and etc. So our headquarters is in Beijing, and we have a sequencing center and a lab in both Germany and China, Qingdao, and we have our bioinformatics center as well in another city of China. So the services that we will provide is kind of, I think it's kind of covering most of the areas that the biomolecular studies that they are doing, like for example, the genomics ones and population genetics, transcriptomics, and microbiome related studies. And we are always want to help more on this biological studies with our like highly qualified services. So thank you so much for listening. And Emma? Okay, so do we have any questions? I saw a hands up, and are we able to pass the? Are we able to pass the? Oh, here.
Okay, cool. Can you hear me?
So sorry, I think. Hello?
Hi, can you hear me?
Yeah.
Okay, perfect. So my name is Arvin, and I'm from the Friedrich Alexander University in Erlangen and Nuremberg. And I'm working on human cancer metastasis, and we use the zebrafish as a model. And I'm curious to know, so first of all, we could actually fit quite a lot of zebrafish larvae on a chip, I guess. So you said that 2 samples would be possible? Would it be possible to fit even more?
As long as it can fit into the, if you remember the cassettes that as long as it can all fit in. And one of the worrying here is that if the tissue is too tiny and we cannot really guarantee they are on the same, say the same level of in the block. So when we are cutting, when we are cutting normally the size of the thickness of the section is around say 10 micrometer to 20 micrometer. And there is another consideration here is that we need to make sure the tissue types that we put in one cassette needs to be pretty much similar to each other. So we don't really need to balance the permeabilization time for one sample to the other.
Yeah, so everything would be the same, and I think generally from our part, getting everything level would be our problem in a sense. But I think in general that should be easy enough, and I would do some H&E staining here before just to see if we could manage. But that would be really cool to check. And then also the last question would be, is it a problem data analysis wise if it's mixed tissue? So this would be the majority zebrafish tissue, and then you'd have some human tumors in there.
Okay. So the pipelines that we have in-house is actually designed for one species only, but we can run two different set of analysis. For example, the data set, we can run it according to zebrafish reference genome first, and then we can run another pipeline with human. Because when we are comparing, we are mainly comparing human genes with human genes. We're not really comparing human genes with zebrafish, right? So it can be in it can be in separate analysis.
Cool. Okay, it looks and sounds really exciting, to be honest. It's really cool.
and again, the only worry here is that if the permeabilized of human tissue is similar to zebrafish.
Okay. So that's something that I have no idea about.
Yeah, it sounds very exciting. It's definitely worth trying, I would say.
Cool. Okay, thank you.
Okay, no worries. And we have another question in the Q&A box, one from Jason asking how to prepare the samples. So it's mainly depending on different sample types. For example, if it is an animal tissue, normally what we do is that we make sure we cut exactly the position or the parts that we want and remove any kind of contaminations. For example, if there are bloods or any unnecessary tissues, we need to remove it. And then we do have a video guidance to show you how to do the OCT embedding, because for me it's very straightforward. It's mainly on the cassette. We put the OCT medium in, and then we position the tissue in the right position while it's cold, and we can freeze the OCT block or the OCT medium in liquid nitrogen with the isopainting while it can caps into the very perfect structure. Yeah, and once the block is completely frozen, it can be shipped in dry ice to the lab, or from our side, then we can perform the QC and also the sectioning. And another question here is from a question is on what plant tissue are suitable for spatial transcriptome RNA. Yeah, so the plant cell walls are really difficult to permeabilize, and we have done some of the trials on very young and fresh tissue, or for example, the very young leaves or the very young stems of this, or young roots, where they don't really have a very robust cell walls on the surface, and we are still able to release mRNAs from plant cells. So yeah, so for now the plant tissues, we are always thinking that say the young ones or the ones that are really fresh are better for spatial transcriptome sequencing. And how to future how do you filter the data to remove the sequences part? So so in the pipelines, I'm not really a bioinformatician here, so I'm not an expert in the analysis part. And as far as I understand, the data will be like once we do the data QC to remove any kind of the low quality data and also the adapter contaminations and etc. We will align them to the reference genome and then make use of this data to do the expression codification. And also here, since we are using this UMI barcodes, like the so for each single molecule they have UMI barcode, and once it gets duplicated, the UMI barcode can tell us, okay, these reads are from one molecule that we captured from the sequence. I hope I answered your question. Okay. So yeah, it seems that's all the questions that we get today. And thank you so much for joining today's webinar. And I hope you have a good time in the rest of the day. Thank you.